Yes, hi everyone, and I would like to thanks for opportunity presented our work here. And yes, we've been working. We got mouse like 3 years ago, but unfortunately, it's still very much work in progress. So since CHAMP1 disorder is caused by one gene alteration, we think that it's a good target for gene therapy. And gene therapy based on AAV currently are in advanced stage. They've been widely used for different diseases, of course monogenic diseases, and there is limitation in gene size. We cannot pack huge genes in there. But for CHAMP, it's a perfect size, AAV, an optimal gene delivery vehicle for central nervous system. It has advantages, such as you can see here, can infect both dividing and undividing cells, display strong preference to neuronal transduction, enable prolonged expression, and show no pathology in data that have been reported. So our approach was very straightforward, just 6 steps: obtain mouse with CHAMP mutation, phenotype it, and we expect to see some robust differences. Identify suitable vector because there is different type of AVs. And the first 3 steps somewhat done. Currently we're working on step 4 and 5 simultaneously, attempting gene therapy rescue and perform phenotype and molecular analysis. The only step that was not touched in any way, it's take to a human, since we need to finish with mice first and there is a lot of regulations. We purchased a mouse from University of California. Mouse has 35 base deletion, which mimics a mutation that was found in one of CHAMP kids. It's easy to genotype. Heterozygote has 2 bands. Homozygote, which is normal control, have just 1 band. And our mice is embryonic lethal, so we never get any homozygote mutant. So we're working with heterozygotes, and based on previously published Tanaka's mice, we expect that our phenotype should be mild, but maybe more severe than their mice. And from very beginning, when we start expanding our colony, we notice that size looks like they smaller. So when we update weight, we can see small but statistical significant difference for both sexes, male, females, and for different time points. We do weight at 3 weeks old when we wean them, and at 13 weeks when we finish our behavioral studies. So we consider it as a good sign because quite often mutant mice don't have any phenotype. So initially we plan to do behavioral tests. You can see them here, and we divided them in cognitive battery tests. You can see here label, and then motor and reactivity battery. And when we start doing it again, we decided that we need to have at least 20 mice per group. When we start doing rotarod, we saw that males and females learning differently. So we have to increase numbers to have 20 mice per group per each sex. And when we finally, almost a year in research, done all this and had enough numbers, we realized that results from these initial studies are extremely variable, and even more, wild-type groups were not typical for C57 black 6 background mice. We performed ordered some primers, performed genotyping, and realized that J alleles are absent in that mice. So what we end up with, unidentified outbred strain background, which would not probably would require huge number of mice, 20, 40, we couldn't assess, and also makes results not reproducible and difficult to interpret. Since our goal was gene therapy, so basically we're looking for difference between heterozygotes and control mice. They need to be robust, reproducible, and statistical significant. So when we treat mice with AAV, we can see differences like improvement in behavior. And there is no such thing as just black 6 mice. They were bred in different facilities for decades and become kind of independent, even if they look the same. They have more than 100 genes, have alteration, more than 100 genes. They different metabolism, behavior, response to treatment, including AAV. So we decided, make hard decision to postpone studies, suspend, and start crossing mice to actual true 6J background. We were purchasing mice from Jackson, and the true congenic background considered after 10 crosses, generation 10. However, we decided to resume behavioral testing starting at generation 4. Okay, so before I present result of that behavioral studies, I want to point out that while we were crossing and waiting, we work with some cell cultures using CRISPR technology, generated multiple clones with different CHAMP1 mutations, and by screening, we were able to find clones, like you see here, the ones that were completely absent of CHAMP1 expression. And it's done in 2 different cell lines. This is just representative image. So clone with complete absence of CHAMP expression was treated with AAV, the one we chosen to use in mice. And we were able to confirm in expression on mRNA level, you can see below, and most importantly on protein level. Expression was detectable up to 72 hours. So again, it was very encouraging because AAV actually designed to work in vivo, in mouse and animal models. They don't really express highly in cell lines. So we were very confident that it will express in mouse model. When we came back to perform behavioral studies, so because we still kind of characterizing and looking for that significant differences between mutant, heterozygote, and wild type, we're doing 2 experiments at once. We're still performing behavioral phenotyping of the CHAMP mutation, and we doing reintroduction of CHAMP construct and checking how behavior, if it's changing behavior. So at every generation, we're injecting and testing. We change our behavioral procedures. Now we're performing only these 5 tests you can see below, but every mouse is goes through these tests. Okay, so far we've done gene expression study for 3 cohorts. It's cohort in generation 4, 5, and 7. So our true congenic background was not tested yet. Currently we're at 9 generations, so we're getting close. And PAPs are injected on day 1. It's intracranial injection, so in brain injection at each size. At day 1, it's mouse head is very soft, so it's easily done. They would just put them on ice and inject. It takes a couple seconds. So all mice, all these pups are doing fine, and we start performing behavioral procedure. Okay, so again, before going to behavioral studies, I want to say that we were able to confirm expression of CHAMP and on on RNA level, like this is on RNA level, and this is PCR results. We can see increase up to 16 weeks. And I have to say that we chose, because again, final goal to do gene therapy in humans, our AAV is it's a human gene. It's CHAMP1 human gene. That's why you see such huge difference. So basically from nothing to hundreds folds increase. But we see increase in spinal cord as well. But remember, like it was injected intracranial, just in spinal cord. We are also able to confirm expression in mouse brain on Western blot. Densitometry shown on other side. So like our AAV is expressed. It's stably expressed at least for 16 weeks. So question now is like, is that treatment improves behavior? First test we're doing, it's elevated zero maze. So we are looking for like anxiety related behavior. And at this point, we're looking for pretty much like any test that will be statistical significant between control and heterozygotes. We have to divide them in male, females by sex, again, because we saw differences in behavior before. So this is wild type males receiving vehicle, heterozygote males receiving vehicle, CHAMP, normal males receiving CHAMP AAV and heterozygote receiving CHAMP AAV. While numbers, unfortunately, is still, you can see number in parentheses, numbers are still small. We see a trend for increased time span in an open area in CHAMP1 mutants. And when we're crossing them longer, like at higher generation, we see that trends kind of increasing. So we expect it to become statistical significant when we have fully congenic. And it might be used at assessment of effectiveness of gene therapy. So next test we're doing, it's Y-maze, spontaneous alteration. Mouse is put like in one arm, and they tend to alternate the arms they're exploring. If they cannot recall which arm they just went, it would suggest failure in like working memory. Again, numbers you see here, so far it's not significant, and we thinking that there is no, for this mouse, there is no differences in working memory. So next one, just like in rats before, we're doing novel object recognition, but we're only checking long-term memory. So after mice familiar with Arena for several days, we introduce like 2 object identical, and 24 hours later we switch one of object for new object. Graphs here just show that like in pre-exposure, mice show typical habitation to empty arenas. So in results of novel object, when we had together generation 4, 5, and 7, it looked like we don't see any differences. All mice show preferences to the novel object, but when we remove early generation, like generation 4, there is trend which we consider as like very encouraging. So far it appears only in males, and gene treatment brings numbers back up. In females, we keep seeing like very huge alteration. So we keep crossing, and our expectation that this will be statistical significant test which we can use to assess gene therapy. Next one, it's a contextual fear conditioning. So mice put in some unique chamber it never seen before, let it explore, and then it receive food shock. So next day, we bring it in the same chamber. If it can make associations that something like bad is happening soon, it will freeze. We don't see, like in recall trials, we don't see differences. So we believe that long-term association memory is intact as well. As to acoustic startle, we checking sensory reactivity, assessing the acoustic startle response. Basically, it's like reflexive whole body flinch or jump, as the people say. It occurs in response to sudden intense noise in our case. Right here, you can see that they just show typical response. As noise increase, they kind of flinch or jump more. Next is acoustic startle pre-pulse inhibition. So before high intensity stimuli, we give it like less intense. And it needs to kind of prepare mice to what is coming. So it shows ability of brain filter out noise and make association. So in our mice, all of them show typical attenuated response. So again, sensory motor interaction is intact. Now to our, I feel hesitant even put conclusion because we have so few mice and we still crossing them, and when we into congenic background, we will get 20 mice per group to make actual conclusion. But so observation, discovery that CHAMP1 line we received with on outbred unknown background caused significant delays, but we're breeding it on pure 6J background. So we will isolate and study just specific effect of CHAMP1 gene on behavior, not just some other gene mutations. As to data of those 5 tests we perform so far, only there is no statistical significant differences because numbers are small. But we feel that that encouraging test toward novel object recognition we see, and that it's resolved in mice after gene therapy. So we will be able to use this test for assessment of gene therapy. As I said before, no differences in contextual fear conditioning. Acoustic response is typical. So again, it's preliminary results, and when we get this final congenic mice, we will offer it to Jackson lab. So everybody who wants to work with these mice doing any behavioral study will be able to do it. So thank you. So there is people involved in Children's Hospital of Philadelphia and University of Pennsylvania. And also I would like to thank you, CHAMP Research Foundation, and of course CHAMP families for being so generous with time and samples. Thank you.
Thank you, Marina.